Good evening. Good evening and welcome to Loyola University Maryland's 6th Annual Grand Seminar Lecture, the signature event of the Loyola University Maryland's celebration of Science Week. I'm Amanda Thomas, Dean of Loyola College of Arts and Sciences. We have been looking forward to welcoming you to our campus today for this chance to highlight our natural and applied science programs. The Natural and Applied Sciences Grand Seminar was created in order to bring speakers and topical subjects related to the natural and applied sciences to the entire Loyola community for students and faculty members from all disciplines to appreciate and enjoy. Each year, the Grand Seminar discusses a topic so broad that not only natural and applied science majors, but also the greater community can understand and appreciate it. The Grand Seminar is just one of the many programs and initiatives Loyola has launched to affirm and advance our commitment to the role of sciences in our university. There is much to be excited about in the sciences at Loyola today, and I'm delighted that you have joined us here for this celebration. Tonight, we are so honored to welcome Dr. Bonnie Bassler, recipient of the 2015 Shaw Prize in Life Science and Medicine and the Squibb Professor of Molecular Biology at Princeton University. To introduce Dr. Bassler, I welcome Dr. Bahram Rugani, Associate Dean for the Natural and Applied Sciences. Dr. Rugani.
Thank you, Dean Thomas, and thank you to all students, faculty, alumni, and guests for joining us this evening. Dr. Bonnie Bassler has established her reputation as a teacher and a scholar who can make science accessible even to people who aren't scientists. There is a reason why her TED Talk, Tiny Conspiracy, Cell-to-Cell Communication in Bacteria, has nearly 2 million views. But it was at a science meeting in Baltimore that she attended as a doctoral student, where a geneticist named Mike Silverman first introduced her to his research of glow-in-the-dark bacteria. The bacteria, when they reach certain cell numbers, all turn on their lights together. It made the invisible world visible, Dr. Bassler realized. She became Dr. Silverman's postdoc and began her study of glow-in-the-dark bacteria that has become the focus of her research. Today, Dr. Bassler is MacArthur Fellowship recipient and the Howard Hughes Medical Institute investigator. She was the 2004 Inventor of the Year by the New York Intellectual Property Law Association for her idea that interfering with autoinducer 2 language could form the basis of a new type of broad-spectrum antibiotic. Autoinducer 2, or AI2, is a unique biomolecular incorporating boron that is known as a signaling molecule used in quorum sensing. Recently, Dr. Bassler was awarded the prestigious 2015 Shaw Prize in Life Science and Medicine. She is a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. She has also received special recognition from the World Cultural Council. Dr. Bassler received her bachelor's degree in biochemistry from the University of California, Davis, and she earned her doctorate in biochemistry from one of our neighbors, the Johns Hopkins University. We are indeed honored to have Dr. Bassler here to address us tonight. Please join me in welcoming her back to Baltimore to Loyola University, Maryland. Dr. Bassler.
Thank you for that lovely introduction. It's good to be home. I haven't been here in a while, and I used to come actually to this campus a lot when I was a graduate student here. It's changed a lot. Anyway, I want to thank you for the generous introduction and also the generous invitation. I have to tell you that the instant I got invited to give this talk, I said yes, because I was like, what kind of a place spends a week celebrating science? That is so forward-looking and so important for this country and for the world and for young scientists and for everybody. And I just want to say that it's so meaningful to me to be able to give this important, nice talk, because I just think that's the nicest idea I've heard in ages. So thank you for this wonderful, wonderful day. It's been magical. It's been brain candy to learn about what's going on here. And now I'm glad to actually do the reverse and to spend a while. I think I'm going to come down here. This looks dangerous. You'll see that for a person like me. Anyway, and so I'm really glad. I'm still sort of your neighbor, just to train right away. And so what I want to tell you tonight is about some of the new work that my gang is doing in the lab at Princeton. Okay, and so I know that there's all kinds of people here, all kinds of scientists, all kinds of non-scientists, and so I want to put us all on the same place and tell you about the critters that I love more than anything, which are bacteria. So bacteria were the first organisms on Earth. They arrived soon after oxygen got here, so they've been on the Earth for almost 3.5 billion years. And if you look at them under a microscope, so they're really tiny, so you have to have a microscope to see them. We can look at them, we can see them, and we can actually draw cartoons of them. And so if you draw one, like this would be, is that working? Actually, I realize, this is it. Okay. If you actually look at what a bacteria, this would be like an E. coli in your gut. All bacteria look pretty much the same, right? So they're single-celled organisms, and they're covered with a membrane that keeps the outside world out and the inside world in. And inside of them, they have the cytoplasm, which has all these biomolecules roiling around, that keeps them alive. And then what's special about bacteria is that they each have one piece of DNA. So they have one chromosome. So they only have a couple thousand genes, so not very much genetic information to give them their physical forms and then any behaviors that they would have. Right? So they're very primitive, stripped down, simple-looking organisms. But what I want to try to convince you tonight is that looks can be deceiving, that they are smart, that they can do tricks. I want to try to convince you, in fact, that they can talk to each other with a chemical language, and that they're really good at math, that they can count their numbers, and that they can act in groups. And the ability to do that actually lets bacteria do many of the magical and horrible things that they do on this earth. And that evolving the ability to have collective behaviors was a huge step. And that step happened in bacteria. And so I want to tell you about that, and then I want to tell you about, and you already heard about this, our attempts to actually try to thwart them from talking to each other to make new medicines. And so that's where we're going to go. And this all started from the idea, what drives my lab is wondering, how do bacteria get any bang for their buck? They're so small, how can they manage to do the things that they know that they do? Okay, and it's all going to have to do with because they can talk to each other. Okay, so to make sure to motivate you to be interested in that topic, well, the doors are closed, so tough you're here. But anyway, just in case, to motivate you, I want to try to tell you something about your relationship with bacteria. And so this is the Bonnie Bassler view of the world. So this is supposed to be my idea of a human being. And I made this cartoon of this man, and each of the circles inside is supposed to represent some number of the human cells. So we know that it takes a few trillion cells to make a human being. So that gives you your form, and it does all the jobs in your body to keep you alive. At any moment in your life, you have 10 times more bacterial cells in you or on you than human cells. So by this metric, you're 10% human. You're 90% bacterial. But of course, this is the 21st century, and we know lots about DNA and the genome, so I've remade my man out of the A's, T's, G's, and C's that are the famous nucleotides of the DNA double helix, right? So the human genome has been sequenced. We know how many genes there are. There are about 20 or 25,000 genes in the human genome. So that's how many bits of information it takes to make you a human being. at any moment in your life, you have 100 times more bacterial genes providing you proteins that do things than you have human genes. So these bacteria give you this repertoire of genes and proteins that do work, and they are essential for life. And so you can pick whichever of these metrics you like. At best, you're 10% human. I think you're really 1% human. The other 99% of you is bacterial. And so this is true for every higher organism, every insect, every plant, every animal, every human. We all live in these consortia with these bacteria, and they are not passive riders. They are supplying you things that your genome doesn't have, as I said, and what we now get is that there would be no life on Earth if it weren't for bacteria, because our genomes don't encode the processes we need to actually be alive. Most of that, or a lot of that, comes from bacteria. Okay, so that is to frame this conversation and why you might want to learn about bacteria. And so now moving a little bit forward, maybe, right? I don't want to be a Pollyanna about this, right? If you've heard about bacteria, they usually get a lot of bad press, right? And so for sure, there's all these bacteria in the world that have no business being in you or on you. And if they get in or on you, they can make you sick or they can kill you. And so historically, for almost 200 years, we've known about bacteria that do this. But now, in the last 10 or 20 years, what we're learning is that they're not all bad, that in fact most of our relationships with bacteria is profitable because they give you all of these properties that you don't have. So for example, they're very important for digesting food. If you eat plant food, if you eat a salad, you don't have the enzymes to get the nutrients and the calories from that. Those are supplied by bacteria in your gut. They make lots of our vitamins that are vital for keeping us alive. We know that somehow the bacteria that live in and on us educate our immune system, and they tell the immune system, these are bad microbes, you should keep them out, but I'm a good microbe and you should keep me in. And so they do all of these jobs that we're only now learning about. Right? So the point that I want to make with this slide is whether you want to think about the harmful bacteria or you want to think about the beneficial bacteria, right? There's both kinds on Earth. And the question that is at the heart of my lab study is no matter which of these behaviors you want to think about, how do the bacteria do this? How can it be that they have us on our knees in this one case, and in the other case, they're actually keeping us alive, given that they are microscopic, right? How do they do it? And that's what my lab tries to study. How do bacteria get a bang for their buck? And so you already heard from the dean that this all started, this idea that bacteria could talk to each other and that they could work in groups. It came from a really anomalous, crazy observation that had to do with a squid. And so what I thought I would do is just tell you, as this is especially for the students, our path of how we got to where we are about these bacteria talking to each other. And so I'm going to just sort of give you the intellectual science path from the beginning. And don't worry, the field's only 20 years old, so it's not that long. Anyway, so it all started with this squid. So this is the Hawaiian bobtail squid. It's about this big, full grown, and it lives just off the coasts of Hawaii in sort of shallow, knee-deep water. And it lives in a very beautiful one-to-one symbiosis with a bioluminescent marine bacterium called Vibrio fischeri. So Vibrio fischeri lives in a light organ in the squid. So the squid gives the bacterium a home, and the bacterium gives the squid bioluminescence. And so this was the first organism where we learned about these bacteria talking to each other. So I want to tell you about this partnership. So will you show my movie, please? Thank you. Okay, so first I'm going to tell you from the squid's point of view. So this animal is nocturnal. And so what we've done is we've put it into an aquarium with the lights on. And so it looks kind of spastic. And that's because it's nocturnal. And during the day, when the sun would be out, it's supposed to be asleep. Right? And so it never likes to be out in the sun. It thinks it's going to get eaten by a predator. So now what we've done, the same squid, it's in the aquarium, and we gave it some sediment. And so the light is on, it thinks it's daytime, and so what you're seeing is the way it escapes from predators during the day. It's thorough. You only get eaten by a predator once. Oh, this goes with tonight's award. Okay, good. Good for me. Anyway. Okay, so now what you see is it's going to retract its tentacles, right? And if I showed you that aquarium, of course you wouldn't know there's a squid in there. There's actually about 10 of them in there. So that is a fantastic strategy if you want to escape from predators during the day. You might have saw it camouflaged itself, and then it buried itself in the pebbles, and of course predators can't see it. Okay, so that's a great solution. The problem is it can't stay there 24/7. At night, the squid has to come out and hunt. It also has to eat. And so that's where the problem exists, is how do you stay invisible at night? Because what I told you is that this squid just lives in this shallow, knee-deep water. So on bright lights, when there's lots of starlight or moonlight, that light can penetrate the depth of the water the squid lives in. So if the squid was swimming around and the moonlight shone on it, it would make a shadow on the seafloor, and then predators could see its shadow, calculate its trajectory, and eat it. So how does the squid manage that? That's where the bacterium comes in. And so now what I've done is the squid is turned on its back, and I hope what you can see are these two glowing lobes that make up a light organ that's under the mantle or the body of the squid. And inside that light organ is this beautiful bacterium, Vibrio fischeri, that has the special property that it makes bioluminescence. It makes light. And so it's in this light organ at something like 10 to the 12th cells per milliliter. It's like a paste of this bacteria. And they're in there, and they're making bioluminescence. And so what happens is when the squid emerges at night and it's swimming around, it actually has detectors on its back. So it senses how much moonlight and starlight it's hitting its back. And then it opens and closes a shutter. It's just its ink sac over this light organ. So it modulates how much light comes out of the bottom, which is made by the bacteria, depending on how much light hits the squid's back. So it actually doesn't make a shadow when it swims around by using this counter-illumination procedure that is given to it by the squid. So this is sort of like the stealth bomber of the ocean, right? It cloaks itself in this invisibility device, and then it doesn't get eaten by predators. So now it has a way to be invisible during the day and invisible during the night. But my interest, most interest in this, is about the bacteria, because it turns out that the bacteria only make light at night. And what the squid does is every morning when the sun comes out and it buries itself in sand, it's got this thick 10 to the 12th cells per milliliter of bacteria, and it can't maintain that forever. So when the sun comes out, the squid has a pump that's connected to its circadian rhythm. So it pumps out like 95% of the bacteria. So now the bacteria are dilute, but the squid doesn't care. It's asleep in the sand. It doesn't need light. And then as the day goes by, the bacteria grow and grow and grow and grow. And at night, they're at high cell number again, and then they turn on light. And so that's perfect for the squid. The light is only on when the squid needs it. But my question was, how does the bacteria know whether it's dilute or whether it's at high cell number, which actually translates to daylight and nighttime? And so that's what I wanted to know about. And so the way that it works, and you kind of heard a little thing about this already, is that the way the bacteria understand what the squid has done to them is because they talk to each other and they count their numbers and they recognize when they're alone and when they're in a group. And they only turn on light when they're in the group. And so now for the rest of this talk, it'll be from the bacterium's point of view. So this is supposed to be my cartoon of a bacteria, of a Vibrio fischeri cell. So what happens is when the squid squirts it out in the morning, and the bacteria at low cell number, right, even in this light organ, the world is big and the bacteria are small. They're at low cell density. So they have some genes turn on that are good for acting as an individual. And among the things they do is that they make and release a small molecule, which is by this triangle in my slide, that you can think of like a hormone. And so what happens is, since the bacteria are at low number, they release the hormone, and the hormone just diffuses away. And so we actually call this hormone an autoinducer. And so it diffuses away, the bacteria can't detect it, and that says, don't make light. But then, as the bacteria grow, and they just grow by fission, they just grow and divide and divide and divide, and one cell makes two, and two makes four, and so on and so forth. What happens is, since all of the bacteria are making and releasing this autoinducer, this hormone molecule, the concentration of the autoinducer increases in proportion to cell number. And when this autoinducer, which is outside of the cells, reaches a critical threshold amount, the bacteria detect it, and then in unison, they infer by detection of that molecule that they must have neighbors around. So in synchrony, they all turn their light on together. So in fact, they can't really talk, obviously, to each other. They use the concentration of this chemical as a proxy for cell number. And when it's at a high amount, they think, if you will, that they have neighbors around, and so they all turn on light. And so this is awesome in the squid, because in the daytime, the bacteria are like this, and in the night, they're like this. And in fact, we know we're right about this, because what we can do is we can grow these cells up, spin them out with the centrifuge, and take the liquid that's left over. And if we take that liquid and we put them on these cells, they will turn on light. So they have no idea that there's other cells around. They're simply measuring the environment for this chemical, right? And so it's a mechanism for them to communicate with one another and say, I'm here. And so then you already heard from the dean that this bioluminescence was this amazing trait because we could just see it, right? And we could follow it and we could measure it as scientists. And so using that as the readout of the cells talking to each other, Mike Silverman and I wanted to understand what's the molecular biology behind this. And so what we found, so this is now again supposed to be the bacterial cell, what we found was that there was an enzyme that makes that hormone, the autoinducer molecule. And that diffuses in and out of cells, right? So the more cells there are, the more there is. And then there was a partner protein, a receptor, that was on the cell membrane. And so this is just like the receptors that receive hormones on your cell membranes. So there was a protein that spanned the outside to the inside of the cell. So it could detect, it could bind this hormone molecule, and when it slots into that little binding site, information comes into the cells to tell the cells to turn on the genes that make the enzymes that make bioluminescence. So it's really simple the way this works. The more the bacteria there are, the more of this hormone there is that tracks with cell number, and then at a critical hormone concentration, which is a particular cell number, all the bacteria together turn on light. And so that was interesting in terms of this squid and the symbiosis, but the reason that it was especially interesting is once we had our hands on the genes, we could start looking in other bacteria. And what we realized very soon after we started was that this was not just some goofy anomaly of this glow-in-the-dark bacteria in the ocean. At last count, there are thousands of species of bacteria that are known to have these little circuits. So probably, we haven't studied all bacteria, but probably all bacteria, it's very useful to know when you're alone or know when you're in a group. So at least thousands of species so far all have an enzyme that makes a signal molecule. There's always a partner protein, and then together these complexes turn on all kinds of collective behaviors that bacteria want to express when they're in communities, but not when they're alone. And so far, so the problem was we just couldn't see it, because these are things like virulence, like toxin production, like biofilm formation, how bacteria sit on surfaces in groups, the release of all kinds of exoproducts. And so what was so fantastic about bioluminescence is we could just see with our eyes the bacteria do it. But then we got sophisticated and we started measuring other traits, and it turns out that typically in any species of bacteria that you measure, typically 600 or so genes will be controlled by quorum sensing. So it's a big deal for a bacterium to go from being alone to acting as a group, and they turn on all of these genes, including virulence genes in the pathogens, that when all the cells are acting together, these behaviors become successful. And so now we have a fancy name for this. We call this quorum sensing. The bacteria vote with these chemical votes. The vote gets counted, and then as a democracy, they go along with the vote, right? And so what has been really interesting, so that's quorum sensing, and what's been really interesting, again, with the field has sort of turned into studying pathogenesis, is what we know is that if we make mutants that can't make the molecule or can't detect them, they are completely avirulent, right? So the idea is, again, from the bacterium's point of view, if some bacterium is infecting you, and there's just a few cells there, the last thing it wants to do is to release some toxin. First of all, because a couple of cells can't make enough to make a success, to make any difference. And also these public goods, these things that get released, those are red flags for the immune system, right? Your immune system evolved to hunt down bad bacteria and get rid of them. So the better idea from the bacterium's point of view is to wait, to count yourselves with these small molecules, to recognize when they have the right number, that if they all launch together, they will be successful. And so now we have dozens of cases of these clinically relevant bacteria that can't cause disease unless they have quorum sensing. Okay, so that's the field. And then, these are not actually triangles, these are chemicals. And so we and other people in the field started to ask, well, what are these little chemical words that these bacteria are talking with? And so, of course, the first one discovered came from Vibrio fischeri, because that's the bacterium we've known about the longest. And so in my cartoons, it's a triangle, but this is the actual chemical that Vibrio fischeri uses to talk. And so then we started to discover them in all kinds of other bacteria, and I've just put a little smattering of different species of quorum-sensing bacteria and the molecule that each of these bacteria talks with. And I hope what you can see is that the molecules are all related to each other, right? So the left-hand part with this ring is identical no matter what species of bacterium it is. But this right-hand part, these are just carbons, is a little bit different in every single species. And what that does is to confer exquisite species specificities to these languages. So each of these species recognizes its own signal and nobody else's. And so they really do fit like locks and keys into these receptors. You know how you put your key, all the keys look the same, but you got to put the right one in the right lock? That's how these work. So what I mean by that is if I take the agrobacterium molecule and I give it to Vibrio fischeri, nothing happens. And likewise, the Vibrio fischeri molecule doesn't work on agrobacterium. So what we understand is that these molecules enable intra-species communication. These are private, secret languages that bacteria carry out with your conversations, that they carry out with their siblings so they can count how many of their brothers and sisters are around. So that's how the field started. And then once we sort of my lab got that far in our studies, we started to think, well, this is really interesting and it's neat if you're Vibrio fischeri and you live in this one-to-one symbiosis with this squid. But most bacteria sort of live in the Wild West, right? They live not alone. They live in consortia with hundreds or thousands of other species of bacteria. So here is a picture, again, taken under a microscope. This is human skin. This is what your elbow looks like. This is a biofilm. You are covered in a bacterial biofilm. It's always nice to have me right after the snacks. Anyway, so let's see. So the question, so this is a biofilm. We're covered in this, right? And what the point from this slide is simply that every different shape is a different species of bacteria. And so the question my lab next wanted to ask was if this is really about counting, if that's what quorum sensing is, how do bacteria count when they find themselves in a mixture of different species? And so we wanted to study that as our next question. And so to do that, we went back to these bioluminescent bacteria that we knew about. So we knew about a cousin of Vibrio fischeri. Its name is Vibrio harvii. It's a bioluminescent marine bacterium, but it lives a very different lifestyle than Vibrio fischeri. Vibrio harvii lives free living in the ocean. So we knew it only turned on light at high cell number. We knew there was a molecule involved. I already showed you that. But we thought, okay, this bacteria encounters all kinds of other species. So how does it really do quorum sensing? And so the reason to show you this slide, besides the obvious cheap propaganda, for the students, this is what the graduate students do, just in case you're thinking about going to graduate school. Anyway, I just want to tell you how we do this experiment. So this is just a flask and some Petri plates with Vibrio harvii on it. To take this picture, all we did was we turned the lights off in the room. And we took the picture from the light the bacteria are making. We're not doing anything to them. We just see this. And so this is bioluminescence. And so what's so fun for us is that we're geneticists, right? So what we can do is we can just mutate Vibrio harvii, put it on Petri plates, turn the lights off in the room, and look for bacteria that are dark when they should be bright or bright when they should be dark, right? It's super simple, and we just pluck those guys out, sequence what's wrong with them, and that's how we found all of the components that I'm telling you about in these bioluminescent bacteria. Right? So we did this mutant hunt in Vibrio harvii, and what did we find? Well, we found that there was an enzyme that made an autoinducer and a partner receptor. We knew that was going to be true. So we found that molecule that I already showed you. But what was interesting is that when we made mutants in this system, this bacterium still had quorum sensing. So we went hunting again, and what we found was that there was a second enzyme that makes a second molecule, and it also has its partner receptor. And then all of that information from both of these autoinducers comes in to tell the bacteria at high cell number to turn on light and 600 other genes. We're simply measuring light. So what we saw was that there were two quorum sensing systems acting in parallel. And so the question, so we got those genes, and we named this enzyme Lux, for Luxor, the god of light, Lux S, for the synthase that makes this second autoinducer. And what we wondered was, next, after we got these genes, is, well, what is the point in evolution of doing this? Like, if each of these molecules encodes the same piece of information, having two systems isn't better than one. And so we wondered, could these molecules actually tell the bacteria something different? We knew that this molecule was the species-specific molecule. Only Vibrio harvii makes this. So what we did when we got this gene, LuxS, was it turns out, you know how we have the human genome? There are thousands and thousands of bacterial genomes. So you can take your favorite gene, put it into a public database that the NIH has, and the database will tell you if any other organism has that gene. So we did that with this gene LuxS, and what we found was that every bacterium that had been sequenced had a highly conserved LuxS gene. So we thought, that's interesting. This seems to be spread across species. So the next thing we did was we actually purified this molecule and asked, well, what is this molecule? And when we did that, what we found was that the molecule was a 5-carbon molecule decorated with all of these oxygens. I love this molecule, but the only thing that you need to know about this molecule is it didn't matter who we purified it from. It was the identical molecule in every species of bacteria. So unlike the molecules I told you at first, that each are slightly different in every single species, this molecule was generic. And so what we thought then is that this is the trade language, or the bacterial Esperanto. If all the bacteria make the same molecule, they can actually talk across species because they have a common language. And then we proved that that was the case. And so now what we think is that bacteria have to be at least bilingual. There are more molecules to find, but at a minimum, they seem to all be built at least like this, in that they have some way to know self, some molecule, and then they use this autoinducer 2 for interspecies communication. And so, in fact, the computation that we think bacteria do now is the following. The first thing we think they do is they ask the simplest question. They ask, am I alone or am I in a community? So they have these receptors on their surface, and they simply scan the environment for molecules, right? Is there anybody else out there? And if they detect some molecules, they'd start turning on and off certain genes. The other thing that we recently showed that they do is that they actually measure the ratios of the two molecules. And then they do different things based on whether they see a lot of the self molecule versus a lot of the other, i.e. the enemy's molecule. So what they do is they actually ask, if I can say that, is it me and my kin that are mostly in this community, or is it somebody else? And remember, I told you there's 600 genes down here, and they tailor this program of gene expression based on whether they and their family members are winning or whether somebody else is actually in the majority, right? And so that is actually how we think quorum sensing works. We think it's a very sophisticated computation where they can actually count both the number and actually tell self from other using these molecules, right? And so there's going to be more molecules to find, but I think that we get the basic principles, right, that they can use a chemical lexicon to actually tell something more than just what the number is of other cells in the environment. They can tell something about species relatedness because that's also embedded in these chemicals. So now you'll remember that I told you that these, and I should tell you that this molecule also controls pathogenicity in clinically relevant bacteria. So the question that the field is turning to now that we understand that bacteria have quorum sensing and we understand that they have to have it to be able to produce toxins and to make biofilms, the two key steps in an infection, the question is whether or not we can actually interfere with that to think about making new kinds of antimicrobial medicines. And so there's sort of two strategies, and I'm going to just go through one. One is that you could try to target this intra-species communication system. So if you went to a physician and the physician knew what was wrong with you, you want to be treated with a narrow spectrum antimicrobial. So the idea would be if you had that there is a need for molecules, antimicrobials, that target very specific pathogens. Lots of times you go to a physician and you're sick and the physician doesn't know what's wrong with you. And so the way we are typically treated is you're treated by a generic, a broad spectrum antibiotic while the physician tries to figure out what you have, and then your treatment becomes more and more narrow. And so maybe another thing you might want to do is to interfere with this system, and if you could make an anti-autoinducer 2 quorum sensing molecule, that could work against lots of different species of bacteria, right? And so we are actually trying to do both of those things, and I'm going to tell you our newest result, which has to do with this system, the intra-species communication system, because that's our newest result. But before I tell you that, I want to take an interlude. because I always get asked this, and I think this is what is this business with antibiotic resistance, and how might an anti-quorum sensing molecule actually be different? Right? So first, let me tell you, so antibiotics are simply chemicals. They are molecules. I put some famous ones here that you have either taken or you know about. They are simply molecules that have properties that allow them to either stop the growth of bacteria or kill bacteria. And so the question is, so why do these molecules kill bacteria? And they actually, every antibiotic you've ever taken does sort of one of four things. So some antibiotics stop, so that's supposed to be the blue square in these. Some antibiotics inhibit DNA replication. So if a bacteria can't make more of its DNA, it can't divide. So that kills bacteria. Some antibiotics, there's three things. Bacteria have to be able to make DNA, they have to make RNA, and they have to make proteins. So if different antibiotics target these three steps in biology. And so any one of these stops bacteria from growing. So certain antibiotics you've taken target those machines in bacteria. There's antibiotics that actually, like penicillin, pops holes in the bacteria membrane. If you pop a hole, all the goodies fall out. That's an antibiotic, right? And then, right, so antibiotics, every one that you've taken does one of those four things. So what's resistance then? So what that is that bacteria, just like we do, when they replicate their DNA, mistakes happen. And so they get fortuitous, just random mutations. Every now and again, a bacteria will fortuitously get a mutation that makes it so these traditional antibiotics can't bind the target. They just sort of change the target. So for example, they have pumps to pump out toxins. Those pumps could get a mutation, so now you give your antibiotic and it gets pumped right out. It's not going to work if the bacteria gets rid of it. So that's one mechanism of resistance. Other is that they change sort of the shape of these different machines so the antibiotics bounce off and they don't stop, for example, protein synthesis or DNA synthesis. They can also have enzymes that are doing particular jobs in the cell, and by accident, if you get a mutation, an enzyme that's supposed to be doing job A instead degrades your antibiotic. And the other thing they can do is that they can get mutations that just make their cell walls tougher, and so antibiotics can't get in the first place. Right? And so what happens is every time a bacterium divides, it gets random mutations. And occasionally, those happen in these very important proteins, and then the bacteria are resistant to the antibiotic. And that is what you've heard about. So how does that play out when we think about giving antibiotics? Okay, so now if you had some, this could be in your gut, or it could be in a flask, or it could be in an animal. If you have just a population of bacteria, they're all multiplying, and fortuitously somebody is going to get a mutation that just by good luck makes it resistant to an antibiotic, whether or not you've given an antibiotic, right? So this is supposed to be this mutant in a population that is resistant because it got some random mutation. So now, if you give an antibiotic, right, most of the bacteria, they're not resistant. They're just regular bacteria, and so they all get killed, right, by the antibiotic. But this guy is impervious because it's got some it existed in the population, but it has some fortuitous mutation that now if all the other guys get killed, only it and its siblings can grow. And so those resistant mutants then take over, right, and now they and all their babies are also resistant. So do you understand that? So that is why antibiotic resistance spreads, is because when you treat a population of bacteria with an antibiotic, you give an amazing selective advantage for anybody who's resistant by killing off all the non-resistant bacteria. And so then now these get out in the environment and farms, and if you don't take your whole course of antibiotics, right? So they just propagate. Okay, so that's how we get antibiotic resistance. And so then the question is whether you can think about anti-quorum sensing strategies, and maybe those could be different. So now what I just told you is that in the traditional antibiotic, you get cell death of all the bacteria that aren't resistant, and you get propagation of the resistant bacteria. So let's just say we could make an anti-quorum sensing molecule. So this molecule makes it so bacteria can't talk or they can't hear, right? We haven't actually changed their growth. And so if some bacterium in this population gets resistant to the anti-quorum sensing molecule, it starts talking, but nobody else is listening, right? So it's like going, come on, you guys, let's quorum sense, let's make these toxins, let's make these biofilms, right? But nobody else is participating. And so you don't get the benefit of the collective, but you also didn't kill off all the non-resistant bacteria, like in a traditional sense. And so maybe by not selecting for the growth of resistant mutants, this could be a new way to think about antimicrobials. And in the tests that we've done, this guy, we can purposely make resistant mutants. It turns on its quorum sensing cascade, and so it's turning on these 600 genes, and nobody else is doing it. And so in fact, this guy actually has a growth disadvantage, right? Because it's trying so hard, but nobody else is working with it. So I think it's an interesting idea to pursue. Okay, so that's the interlude, and I think everybody knows we have this global problem with antibiotic resistance. There's been no new antibiotics for 40 years. So my lab and others want to try to think about this in this new way of sort of behavior modification instead of growth, no growth. Okay, so now I'll just give you one example to finish this talk. So we decided to try this strategy, and so what we picked was this organism called Pseudomonas aeruginosa, which is a notorious pathogen. So Pseudomonas aeruginosa just lives in the soil. You and I encounter it every day, and it's harmless unless you're immune compromised or you have cystic fibrosis. So people who have the disease cystic fibrosis have a human genetic mutation so that they can't clear their lungs. So you and I breathe in all kinds of gunk, and we have mechanisms to keep our lungs sterile. People who have CF can't do that, so they're permanently colonized with a polymicrobial infection. At a certain point, typically when a person is in his or her teens, for reasons that we don't understand, they become colonized by Pseudomonas aeruginosa, and that's what kills people that have cystic fibrosis. They die of a Pseudomonas infection. And that is because Pseudomonas has quorum sensing. It sits on the lungs and it makes this biofilm. It covers itself in this matrix that's like a suit of armor that's controlled by quorum sensing so that antibiotics can't touch it, and then as a group, it releases 50 or 60 toxins that chew up and damage the person's lung tissue. It's just trying to get nutrients, but the outcome is that these people have terrible lung disease. So it's notorious for that, for people that have burns, it is the highest cause of hospital-acquired infections, right? Because it also makes this thing called a biofilm, which is how it sticks to surfaces. This is a really sticky bacteria. So now so it's all over these hospital surfaces. And so now when you go, and you're going to go get your, your gallbladder removed, what happens is that you have Pseudomonas in the hospital, and it's on all these implanted devices like catheters, like intubation tubes, like surfaces, and so it's sort of running rampant in hospitals. So this is the bacterium that in developing countries we need to be able to think about. And what's lucky for me, right, is that it has quorum sensing, right? And so it has one of these intra-species communication systems that controls biofilms, so how it gets on these surfaces, and then also the release of all of these virulence factors. And so the question that my lab wants to ask is, can we make an anti-quorum sensing strategy, and that could be thought of as a new medicine? And so I'll tell you the steps so far. Okay, so this I showed you on one of my first slides. So I'm talking about this intra-species communication system. So this is the molecule that Pseudomonas uses to talk. It actually has two. It has one that has 4 carbons and one that has 12 carbons. So this is its quorum sensing molecule. And so what we did was we just screened for molecules that inhibited this molecule from interacting with its receptor. And here is our favorite anti-quorum sensing molecule. And I hope what you can see, so remember, this molecule fits like a lock and a key in its receptor, right? And so what you can see, our molecule, this left-hand part, looks just like this, except this oxygen has a sulfur, right? So it's kind of the same shape, except it has this big bump on the right end. And so what happens is when our molecule slots into that receptor lock, it slots in there and it jams it. And so information doesn't come into the cell, but this molecule also can't bind, right? So it's competitive. This simply gets in and blocks the real molecule from doing what it's supposed to do. Okay, so now we know we can make that molecule. We made that synthetically just by thinking about what that one looks like. And so the question is, can this work? Okay, so remember I told you that Pseudomonas' strategy is to first make a biofilm and then release its toxins. And so what we can do in the lab is ask it to make a biofilm. So this is just a Petri plate, and what we do is we just put a little drop of Pseudomonas right in the middle of the Petri plate. And then what you can see is it swarms out, right? It's trying to cover the surface, and it makes this beautiful, to my eye, that's a biofilm. Okay, so it swarms out over the surface as it grows. So if we put into the Petri plate that anti-quorum sensing molecule, no swarming. Okay, so the bacteria grow where we put them, but they can't make the biofilm. So that was actually really good news to us that made us think like there's merit to this idea. So it stops biofilm formation. Okay, so that's step one. The other thing is that these bacteria release these toxins. So we wondered, can our inhibitor repress toxin production? And so Pseudomonas, if you just look at it, this is the normal virulent Pseudomonas in a test tube. It's green, and that's because it makes a toxin, the thing that kills lung cells. That molecule is called piocyanin, like cyan, and it just turns out that it has a green color. So Pseudomonas at high cell number makes a lot of this toxin, this lung-killing molecule called piocyanin, so the whole culture turns green. If we make a quorum sensing mutant, what you can see is the culture's colorless. So that shows you that quorum sensing controls production of that green toxin. So if we take the wild type cells, but instead we put our inhibitor in here, what you can see is we can shut it down like 90%. Right? So it's almost colorless. So now we think we're in business. We've got a molecule that stops biofilm formation and it stops virulence factor production. So then the next question for the lab to ask is, can this actually work in an animal? And so here's the last experiment that I will show you. So we have an animal model of Pseudomonas infection. And so what we do is we just measure how many animals are alive over time. Right? And so, of course, if we don't give any Pseudomonas, the animals just live. I mean, these are just regular animals, and they'll live for a long time, right? If we infect them with Pseudomonas, what you can see is all of the animals die in a day, right? So that's the model. So now if we do this experiment, but simultaneously we give this anti-quorum sensing molecule, what you can see is we can save the animals, right? And so we actually now think that there really is merit to this idea of trying to interfere with behavior. This is not a medicine. You know, this is a molecule we got out of a screen, and we did a little bit of medicinal chemistry. You would need to take a pill this big, and it would kill you, right? And so anyway, we have to now we wanted to get some proof that this was a good idea, and so now what we have to do is refine that molecule. The chemists have to do this and make it really potent and make it safe. And I don't know if we can do those things, right? But we're going to try, and we're also going to hunt for other molecules based on these data. We think that maybe we ought to try to pursue this line of experiments. Okay, and so that is where we are right now. And so I want to tell you just our next step is what I first just said, is we have to make that molecule medicine-like. So medicinal chemists can build in features onto the molecule that make it so it doesn't all go to your kidney and you excrete it, you know, in an hour, right? And so that it's not toxic, and it has to be much more potent. None of that has been done yet, but we're working hard on that. Then the other thing, remember I said that Pseudomonas especially loves to live on surfaces. So the other thing, we've teamed up with Howard Stone, who's an engineer, and engineers actually like to make things. And so what he's trying to do, we're trying to embed our anti-quorum sensing molecules or to link them onto materials. Like, so maybe you could make infection-resistant, you know, heart valves or catheters, or you could make surfaces in the hospital like sinks that are coated with anti-quorum sensing molecules. So we think that's an interesting thing to think about. And so we're trying to do something like that. And then, of course, the other thing I'm just telling you this one story about Pseudomonas. We work on lots of different pathogens in both of these molecules, so there's other pathogens to target. And then the real thing is to do this sort of puzzle that I gave you, which is to actually ask, does this give a molecule like this a longer functional shelf life? Is this really useful in terms of thinking about resistance? And we don't know the answer to that. So that's what we're doing in the applied side. And then what I'll just finish with is to just tell you everything I told you, but in less than 1 minute. Bacteria talk to each other. They don't have voices, they have chemicals. So they use chemistry, we think, as their lexicon. We think there's a lot of molecules out there to find, and these are the first couple. I hope what you think is that by having quorum sensing, what that allows bacteria to do is to be multicellular. Bacteria have been studied for 400 years, and they were always thought to be these primitive asocial recluses. What I would argue is that, in fact, they do act like multicellular organisms, and hopefully, by studying these collective behaviors in these bacteria, we can learn how collective behaviors work in higher organisms, because up through evolution, many of the principles will be the same. I showed you two molecules, and I've argued that that allows bacteria to distinguish self from other. I would argue that's what happens in your body every day. Your heart cells do not get confused with your kidney cells because they use different chemicals to take on specialized jobs and to do particular functions. That is what these bacteria are doing. And again, we hope in the basic side of the work, by studying these things, we're going to learn how division of labor happens, how collective behaviors happen, and how, in fact, cells can take on different fates. We think those principles are also embedded in these ancient bacteria. And then on the applied side, there's a lot of work in the field to develop these strategies to impede quorum sensing. And so I told you one, but I want to finish this by saying, that may be the wrong thing. Remember how I started my talk? Is that these bacteria, the good ones, are keeping us alive. And so maybe we should stop fighting against the bad ones. And if we could learn enough about these commensal bacteria that live in us and on us that absolutely have quorum sensing, maybe you could make their conversations better. And so maybe the real way forward in medicine is pro-quorum sensing molecules to make the good guys better at fighting off the bad guys. We know that these bacteria that live in and on you work hand in hand with your immune system to keep invaders out. And so we and lots of other people are, of course, we don't know so much about those bacteria. We're just learning about these beneficial ones. But we and others are trying to make these pro-quorum sensing molecules as well. And then the last thing is to end with a confession, which is I didn't think any of this up. Once we started to think about, well, maybe we could, manipulate quorum sensing on demand, we said, well, these bacteria have a 3 billion year head start, right? And so if we want to do it, they out there, they live in these really competitive environments, surely they are already trying to thwart one another's quorum sensing systems. And so we and others have been out there digging up dirt and getting seawater and getting gunk off of everything. And sure enough, all of these, both pro and anti-quorum sensing strategies already exist. Like they eat each other's autoinducers. So this guy miscounts because his autoinducer disappears. They make antagonists. They eavesdrop. They free ride. They do all of these shenanigans, right? And so those strategies that we can find in nature, those have won over billions of years of evolution. So the idea that Bonnie Bassler and some 22-year-old chemist, even if she was an undergraduate here, are going to make a better molecule is ridiculous. And I don't care what the chemists in the chemistry department try and tell me. So what we should really do, right, is actually take those strategies and bring them into the lab and tinker with those. But those should be the start, the building blocks. And so there's lots of people have a pony in that race, and I think it's a really exciting field of exploration. Okay, so that's me, us, but of course it's not me. And so especially for the students in the lab, what I want to tell you is that I get the pleasure and the honor of getting to come and give this talk, but I did none of those experiments with my own hand. I'm a typist, okay? But these guys, this gang, which to the students, I hope you see that they look a lot like you. They're all within 5 years of your age, right? These guys thought up every idea that I got the privilege of telling you, and they did every experiment to show they were right. And so being a scientist, it is the greatest life in the world because you get to, go on these adventures and think about things that no one has ever thought about in their lives. But the best part is the people part, is that I work with these people who don't believe what they read, and they think there's more out there to discover, and they're really smart, and they're really agile, and they're really creative, and they just don't have any boundaries, right? And so if you guys, the students, want to talk to me after, or if you want to email me, I just want to encourage you to stick with science, right? Because it's really a wonderful life, and whenever you hear something that's brand new to you, I guarantee you it was done by somebody who's within a few years of your age, not a few years of mine. And so anyway, so this is my gang. I'm really proud of them. I want to thank you both again for having me. It's such a delight, and I thank you for listening. And I think we do questions. Yeah.
Dr. Bassler, it's great that you're doing stuff with prevention and with cure. Are you using any of this stuff for detection so that you can just take a little bit of a fluid from your body and detect which quorum sensing thing it is? And
right. So I thank you for that question. I think there's a huge way forward in diagnostics, right? And so people are trying to make like little dipsticks, that would make a readout if one of those molecules was there, and you could imagine that in the food industry, in the hospital setting, in agriculture, and so people are trying to do that. But remember, we only know a few of the molecules. So the more molecules we know, the more you could actually, you could imagine some big palette of these, dipsticks that would tell you who, good or bad, who's there. And I think there's a lot of work being done in that area.
You just put the fluid in a mass spectrometer and...
Yeah, right, and read it out, right? And so people want ones that they can take out in the field, so like a dipstick kind of thing that makes a color or something. But for sure, right, we can make those molecules. They're all very simple molecules. You saw that. We make them by the bucketful. We know exactly their mass spec, and so you can imagine those kinds of very quantitative measurements for the molecules that we know. Yeah. Yep. Mm-hmm.
Hi. It was an amazing talk.
You're
and
lovely.
My first love is physics. I'm actually a physicist. And I was thinking of some interdisciplinary things that just began lighting in my head. So first of all, I'm thinking of these things as the social insects and the work of E.O. Wilson. I'm wondering if there are analogies there. And then also in the field of computer science, a lot of this kind of nearest neighbor game playing, but also the work that Alan Turing did on chemical gradients as a sophisticated way to do that kind of sensing. So I'm just wondering if you could comment.
I'd be
briefly on
delighted.
that.
And then there's another question.
Just throw in a couple of other things.
Okay, but we got to do these two at a. Okay, they're good at math, but I'm like two at a time. So let me do those first. So the first question is, okay, so I love this idea. So you guys know that like like bees and ants, they have these social structures, and that is driven by chemicals, right? And so so our molecules that I showed don't seem to be in higher organisms, but the closest so you can take the same way you can put your gene in the database, you can put your molecule in a database. The closest molecule is a pheromone in an insect, right? So the idea that perhaps there's either one gave rise to the other, or these are similar sort of solutions because they're good solutions. I love that idea. And for sure, in the bee community, they are talking about quorum sensing, right? The idea that these bees do these jobs at different numbers. So I think that is a matter of time before there's a real link between these two fields. But it smells like it, right? And then the second thing, Alan Turing won World War II, but his best work was in biology. And that was to show, World War II, this was to show that these that how, when you release these molecules, how you can get gradients and how you get information out of them. So I think that work is beautiful. Obviously, it's beautiful. So we do work with a condensed matter physicist, Ned Wingreen, who models the systems, like what I gave you what I was nice enough to do was to not show you this, you are a physicist, the 7 feedback loops that amplify, like if they see one signal, they pay attention to that. And so there's all these beautiful engineering principles under that big arrow, which tune the system, which make the system robust, which make them be able to distinguish better input, output. And so the physicists, they're like, and so they are modeling the system. And then what's wonderful is so they make these ridiculous models, no offense to my host. And then we say, they say, without this feedback loop, it's going to be slower. And then we say, okay, we'll make a mutant and see if it's actually slower. And so we do this very iterative physics genetics with this colleague at Princeton, Ned Wingreen. And that has been fantastic for the biologists trying to understand what are the design principles in that circuit, because it is how engineers and physicists think. That has been, I have had a 20-year collaboration with his lab, and then the students, some of them are physicists and then they do wet biology, some are wet biologists and then they do differential equations, and I think that is the future. Yeah.
Fabulous.
Yeah, it's a lot of fun.
Yeah, I was thinking of a NAND gate.
This is an AND gate. Actually, there's a feedback loop that you can never turn it all the way on or all the way off unless both are there or both are gone. Yes, you got it. Yeah.
My last question is something I am confused. I'm really confused about. Just one thing I'm really confused about. What I understood about viruses is this idea of species jump and that the virus will kill you if you haven't sort of acclimated evolutionarily with it. But you indicate that the bacteria like to toxins and
no, you have it. You have
So-
it right. I don't think a bacterium ever wants to kill you. Right? What they want to be is in the gut. They want to be these commensals. And so when you're alive today and you're in this 100 years, there are these species of bacteria that live out in the world, and they have these bells and whistles, which we would call toxins, for doing whatever their business is out there. And then when they infect a virgin population, we haven't ever seen them before. So our immune system isn't beefed up, and they haven't tamped it down. And so right now, you die of these set of pathogens. 100 years from now, it's a different set, right? And we know very clearly travel, changes in the environment, changes in technology allow populations of bacteria that just, like the great example is Legionnaire's disease. That guy just lives in a pond. We make air conditioning that have these water in the bottom. Those old guys turned it on. It aerosolized the bacteria, and here's this bacteria that nobody had ever been infected with before, right? And so they're not trying to kill you. They really want to work toward this mutualism. Right, that's why like now, these mumps and other things are childhood diseases, and eventually Pseudomonas will be that, just not now, right? Yeah.
Well, thank you again for a wonderful talk.
Thanks.
Suzanne started by saying that her first love was physics, and my first love is biology. And I want to thank you for letting all of us here know that microorganisms are the key to the world, because I've been saying it for a while and nobody listens to me.
Yeah.
So I was struck a little bit by, one, the receptor, right? Is the receptor constitutively produced?
It is not.
It is not.
So the way it works, right, is that they put a little bit of the receptor out on the membrane, right? And then when they detect the molecule, there's a loop to the receptor gene that puts more of it there. So in fact, then they actually expand how well they can detect the molecule, because they sort of put more ears out, right? And so what happens, right, is if they see one of the molecules and not the others, they induce that receptor. So it's like they pay attention to that signal and they ignore the other one, right? So they're amplifying it.
Are there any cell surface molecules involved?
Well, the receptors are on the cell surface. Yeah.
The receptor is, but for instance, via direct contact.
Well, the molecule directly binds the receptor. The autoinducer, that is a binding event. And then the receptor, if you really want, dimerizes and sends information into the cell.
Okay, so I was struck a little bit by the fact that the organism has the ability to recognize self and other, which it just really seems very similar to what happens in an immune response when you have, say, a T cell and specifically, well, let's say an APC, an antigen-presenting cell. Sorry, but I need to know an APC and like a T helper cell, and TCR recognizes foreign antigen, and CD4 recognizes self class 2 MHC, and so there's a recognition of self and other, but they're via two molecules that are directly in contact with the cell always.
Yeah,
And
I think so these are diffusible molecules, the autoinducers for sure. And I mean, there can be parts that we don't know yet, right? I still have a job when I go home tomorrow, right? Anyway, and so I don't know. So the way we understand it is very like that cartoon, that the autoinducers are diffusing and the receptors are trying to detect a concentration threshold. The receptors are there, but they, as far as we can tell, like the autoinducer 1 and the autoinducer 2 receptor, they never interact with each other, as far as we know. So maybe it's a different solution, and maybe there's just parts that we haven't discovered yet, too, right? Yeah.
Hi, Dr. Bassler. It's an honor to see you in person. A lot of my microbiome professors have cited your papers and your TED Talks, and I think it's such an honor to see you.
Oh, you are. This guy has a career. I'm actually better on paper.
Right. So one of my questions is there research on quorum sensing in probiotics found in yogurts, and if there are any, if quorum sensing could enhance the effect of yogurts on gut health?
Right. So the part B, that's the fantasy, right? Did you guys hear? We have this microbiome, and you get it on the way out of the birth canal, and it stays with you forever, right? It's really hard to perturb. And so the evidence that actually any probiotic works now is none, right? Yogurt is great for you. It's filled with calcium, right? And I think if you've had a lot of antibiotics and you have problems, then I think you can make a dent in the microbiome, right? But I think that most of that, that what's so magical and actually fantastic is that stuff we eat, yogurt or anything else, does not perturb, as far as we can tell, your microbiome. That's good, so we don't get sick when we eat bad stuff, right? And so people are working, I think yogurt, probiotics, that is where this microbiome business and hopefully quorum sensing, right, that is where the scientists are going to make probably one of the first big breakthroughs, which is how can we give a bacterium that you basically eat and get it to stay. So what happens is when you eat yogurt, you see them there, but if you stop eating yogurt, then they go away because your normal one takes back over, right? And so anyway, so I think that there's no evidence. So what I should say is we know that the bacteria that are in yogurt, they absolutely have quorum sensing. In a test tube, we can show that. Others have shown that, right? Whether or not we can really deploy that, in like food or medicines, that's the holy grail in this field, right? And so I think that it's going to take people, in my field, quorum sensing people working with the microbiome field, like the mechanism people with the people who really think about who's where and what are they doing, working together, and that is starting.
Thank you.
Thank you.
Hi.
Hello.
I'm a freshman student. And
Oh,
I just
good
want
for
to ask
you.
a very general question. Because quorum sensing is communication through bacteria, I just want to ask, is there a potential of self-mutant cells, for example, like cancer, through quorum sensing can have an inhibition effect?
Wait, so say it again. So that through quorum sensing, like in bacteria cells?
Yeah. Is it the same as like self-mutant cells? Like, for example, if there's cancer cells in body through quorum sensing, what?
Yeah, so I think
Do you think there's a potential in it?
Yeah, I think so. What I will say is, so sure. So the answer is yes, right? And I think that for sure the bacteria get mutations and just randomly, and then you get guys that sort of can't hear or don't. These are called cheaters, right? Or don't talk. Like if I were really going to design the system, I'd make the autoinducer, but I'd make U-turn on all 600 genes, right? And I would just take it up. So I think there are examples of that. So there have to be little molecular police too, right? And then this business with cancer, right? Like whenever the people. There's no data on this, but it's just striking to me. When the cancer biologists draw their pictures, they have cells and then somebody gets out of hand and you get a tumor and then cells leave and they metastasize and go somewhere else. That is exactly what happens in a biofilm. The cells sit on the surface, they grow to a certain size, and then somebody leaves and goes and makes a biofilm somewhere else. And I just keep wondering,
like
those
spreading of cancer. Quorum
is
sensing
it
would
like
be the
that?
key.
Yeah. Are cancer cells counting, right, and doing it, or are biofilm cells using the same rules that metastatic cells use? There's no data for that. It's just that in these two different fields, we draw our cartoons identically, right? I just think that's interesting. So I don't actually know the answer, but I think there's potential for all of these kinds of things to be in eukaryotic cells for sure.
But thank you very
Thank
much.
you. That's awesome that you're a freshman and you asked a question. I would not have asked. I won't even ask the question now. But yeah. But I think that's a testament to Loyola. Yeah.
Hi. My background is computer science, and I see most of what you talk about through data-centric sort of vision. My question is that in the beginning of the talk, you said that, let's say, like, you extrapolated out information to show that in terms of data, the human being is about 1%. If you're correct about this extra data layer of extracellular communication, would that make it, say, 0.1%? Or does that increase
Right.
exponentially
Yeah. Okay.
the data
Yeah. I
layer?
don't know exactly. I don't know the answer to that. So by genes, you're 1% human, right? I want to be really careful. By weight. These are small. You know how you can't lose that last 5 pounds? That's because it's bacteria. You can't lose that last 5 pounds. Okay, so I don't tell that because it's not good for my storyline. But anyway, right, so the cells are 500 times smaller than a human cell, right? So by weight, you're mostly human. By cell number, you're mostly bacterial. By genes, you're really bacterial, right? But your question is then you can make all of these chemicals. That's interesting, right? So do you put this other...
From like an information system
Exactly.
perspective.
I think that's an interesting way to... I've never thought about
Is
that.
this
I
like
think
another data layer?
I think so. Of course I think that, right? Yeah, I think these chemicals are doing all kinds of things. And so you're right. Like how many chemicals can that set of genes make, right? And then how many play out on your body? I think that is one way to think about it.
So
Absolutely.
It's like a metadata or something.
Yeah, well, you're supposed to be thinking about yourself as a meta being now, right? You are, right? That's what my field wants you to do, okay? Right? And so I think that the data and the biochemistry, which you're calling data, I call those chemicals, right? I mean, I'll just give a couple of examples. Like, so for example, when you take particular medicines, depending on what bugs, microbes you have here, they metabolize them to different products. And those are the things that give you bad reactions or make the medicines more potent, right? And we don't understand that. But this idea that there's this repertoire of things going on that ultimately end up in your view as my view as chemicals and yours as this extra layer, that is happening. And I don't know how big it is and I don't know how small it is.
So we don't have necessarily the scale of this quite
well,
yet.
the sort of rule of thumb is each gene is one molecule, right? So it could be it's just one more layer of 100x, right? But molecules interact and you know, so I don't think we know the scale of that. It's a good way to think about it.
Okay, interesting.
Yeah.
Thank you.
Yeah.
So given everything you do with genetics, I'm wondering, do you guys use CRISPR-Cas9? And if so, how has that made an impact on your research?
So
going forward?
CRISPR, if I may, CRISPR-Cas9 is what you're hearing about in the newspaper. It's genome editing, right? So CRISPR-Cas9 came from a bacterium, right? And so we actually don't. So all of those. So we actually don't use CRISPR-Cas9 because the bacterial geneticists already had that trick, right? And so what CRISPR-Cas9 has been so awesome is to be able to do the tricks that bacterial geneticists, we can move genes around, we can put genes in, we can take genes out. That bacterial geneticists have been able to do for almost 100 years, right? Because bacterial genomes are easy to manipulate with restriction enzymes and sort of old molecular biology techniques. CRISPR-Cas9 is really awesome if you're working in anything that's not a bacterium, because everybody else, yeast on up, have not been able to do those nice tricks that have been in the sort of bacterial geneticist handbag for a long time. That's why so much work was done in bacteria originally, was because you could do those tricks. But Jennifer Doudna, who discovered that, she's one of my dearest friends, and she should be the 7th speaker next year, because she's the one that was studying bacteria and wondering why do they have that, and then made it into this genome editing tool. Yeah, but so everybody else in my department uses CRISPR-Cas9 because they work on eukaryotes. Yeah, that's a great question. Yeah.
Well, in the interest of time, I think we should stop the questions, but I want to say this one. I don't remember who said that one, but said that there is no art without facts and there is no science without fancy. And you proved that there is both art and science based on your talk. I think it was a fascinating talk, and we really appreciate that you took the time. One thing I want to just say that I mentioned that Professor Bassler is a teacher and a scholar, and one thing most of you may not know that she had an appointment. I think they're teaching a course this morning, and she's teaching another course tomorrow morning. And I'm really so impressed that she took the time to come here, and she's going to take a train back at 10:50 tonight to get back to Princeton. So
it might not be that good of a class tomorrow morning.
Yeah, I cannot imagine that you're going to have a bad class. It's
you're
impossible.
Sweet.
But so please join me to thank
you
Dr.
for
Bassler.
having me.